that it's okay. So you can see that cBioportal is actually implemented across the world, and currently there are 74 instances. And the cBioportal.ca, which is the Princess Margaret Cancer Center's instance, is also one of them. And our webpage is cBioportal.ca. So the difference between one of these instances and the .org version is that, in speaking for the .ca version ourselves, is that we have access controlled. So you need an account to log in. And once you have an account, if you would like to access certain data sets, you would have to request access permission from the PI. And once you gain the access, then you are able to see these data sets. So they are not public. Whereas on the .org website, it is all public data sets from TCGA, Target, CHOP, all the big consortiums and centers around the world. So today we will be going over cBioportal.org, and don't worry, what you see in these private instances and the public instance is the same way that you would analyze data. So the way that you would see how the mutations are displayed, the structural variants, it's all the same. So you're not missing out on anything. So let's dive in together. So if you could go on your laptop or follow along with me, go to cBioportal.org. And once you go onto their page, this would be the landing page. So let me just zoom in a bit. So there is a lot of information to be seen here. So let's go through this a little bit by little bit. So you can see on the left-hand panel, there's these blue categories. So you can see at the top, they try to organize it in such a way to help the users and guide them into certain data sets to help filter down, because you can see there are a lot of data sets. So at the top, we have pan-cancer, immunogenomic studies. And if we go further down, you can see that these are tissue cancer types. And so for those that don't know, there is an cancer type ontology that the MSKCC group has developed. And so this is what I'm talking about. So if you could just type in OncoTree on Google, it'll bring you to this page. And you can see here, it starts off from the tissue and expands out to these primary tissue types. And if we could just click on any one of these dots, it will further expand into deeper and deeper cancer type details. So going back to here, you can see that on the left-hand side, these categories reflect the same categories here. And so pretend that we are only interested in kidney data sets. So when you click on that, it will filter down all the studies for just kidney. And so you see this blue subsection, that is actually, sorry, let's go. If we go to click kidney, you'll see that the first section is renal cell carcinoma. And then these further black headers are the further cancer type details that you would see stemming from RCC. So there we go. So this is a way of cBioportal helping the users and helping them guide them into specific cancer types. So let's dive deeper into how we look at these. So it's the cancer type, as specific as possible of the cancer type. And then in brackets, it is the center where this data is generated from. So here we have TCGA, and in after that, we have where they published their paper. So then we move on to the right-hand side. This is the total number of samples within that project. And then there are three icons. So the eye, if we hover over the eye, it gives you a quick information of what this project is about. Then we have this little blue book. If it is a gray book, that means there's no publications yet for this project. And if it is blue, it is a publication available. And if we click on it, it'll bring us to their paper. Going back. So then we have the pie icon. So if we click on that, that will just bring us to the study. So we'll get to that in a sec. I just wanted to show you. So this is one way to see the list of projects. Sorry, maybe I should expand this. This is one way to see the list of projects. There is another way to explore the data sets. At the top hand panel, you'll see this data set. So if we click on that, it is the same number of data sets, but configured in a different way for the users. So you can see right away next to each of the project names, there is this download button. So you can download the whole raw data sets for each of these projects because they are public data sets. And then there's a reference. So same idea, it references, it goes to the paper. Then we have, Then we have all these different columns telling us how many samples within this project, how many samples have mutation, or if there are any RNA-seq data. And we can go on this columns button and we can select them all and we can reconfigure to see how many projects actually have protein mass spectrometry. And so we're like, oh, this one looks interesting. So then we can go and explore there. So going back to the landing page, how we get there is we can click on this icon on cBioportal at the top left corner. Oh my God. Sorry. So we click in there and we'll come back to here. So let's start exploring the project. So we can type in, we can also search for maybe all the projects from TCGA and it'll filter down the projects. So let's go explore this one. So we can just click on the left-hand check mark next to the project and click explore selected studies, or we can click on the pie. So go there. So this is what we call the project summary page. So this is has a lot of information here. So at the top, you will see in bolded that there's the total number of patients and the total number of samples. Then there are many tables and charts here. We will get to that in a sec. First things first, you'll see the project name at the very top, and then there's a download button again for you to download the raw genomic and clinical files. There's a download button on the right-hand side here, and this is just to download the clinical data. Then there's a little description of what the project is about, and again, the PubMed link to their paper. Then we have the clinical data tab where you can search for the IDs if you want, and this displays all the clinical data. Of course, this is not everything that's available. If we click on this columns drop down, there are all these clinical data attributes as well, if you want to see that. So we can readily just click on any of these patient ID links or sample IDs, and that will bring us to the patient page. But of course, going back and forth, this is a bit cumbersome. Say you're like, I want to cycle through each and every patient for this specific study. So we can do that by going to this little person icon at the top. So if we click on there, we will see now that there's these navigation arrows at the top right corner. So now we can cycle through and look at every single patient if we wish, or the double arrows to go back. We can also, I'm not sure if you can see this little box, you can type in the 10th patient, click enter on your keyboard, and it'll bring you to that 10th patient. So going to the beginning. So let's dive into deeper into how the patient page is like. So at the very top, you'll see that it is the patient and then the samples row. So within the patient, there's a patient ID. And if you hover over this ID, we have this little pop-up box and it tells you all the information about the patient. And then it has some metadata about the patient. And again, what we looked at earlier, the oncotree, you can see that this patient has breast cancer, and then in brackets, more specific, what type of breast cancer. Then you will see a row of all the samples. So say this patient has 10 samples, then all the samples will be listed on this row, and there are specific sample IDs. And when you hover over each of these sample IDs, you will also get the sample attributes. And earlier we mentioned the oncotree code. So here it is, and with the cancer type and cancer type detailed. And of course, this is a little bit cumbersome to look at. We can also go to the clinical data tab, and all that information that we saw in those pop-up box are listed static. And so this is an easier way for a user to look at this information. Going back to the summary tab within the patient page, we can see that there's this timeline here. So this is a quick snapshot of the whole patient's journey from the date of diagnosis, which is timeline 0 up top here on the left side, and all the way to 10.5 years into their diagnosis. So we can see, let's just collapse this for a sec. We can see that this patient had a surgery at the beginning. If we hover over that dot, we'll get more information on the type of surgery and their details. And then we see that they had treatment. And so within each of these treatment, we can hover and also see more details. Now, if we expand it, we can see that it's indented. So we can see that this patient had different lines of treatment, and each of these lines is something different. And further indented are the drugs, or as cBio terminology, they call it an agent. So we can hover over each of these timeline points, and there are different information. And again, in the clinical tab, if we scroll to the bottom, all that information is here for the user to view statically, and you can download each of these sections and search through it through these search fields. So then in the summary tab, if we move along down, further down, we can see this is the section. If we hover over the information, it starts with the genomic data. So this is where we can see some of the segments and the copy number alterations. And so if we hover over each spot and click on it, it will show us the sample ID and some of the stats on the log2 values and the location specifically. Sorry, to reference back here, you can see that sample 1 has a black dot and a 1 and 2. So that means any of these and any entries here, it will be pertaining to that specific sample. So let's go to the mutation section just below. You can see that all of these variants are for sample 1 has it, whereas sample 2 does not have any of these. So we can see that there are some icons. So let's go over that. So at the very top underneath annotation, there's this blue circle. So that is for OncoKB, and that is also affiliated with the OncoTree that I mentioned earlier. So those codes, each of these cancer types in brackets, there's this acronym. So that acronym is what we call the OncoTree code. So going back to here, these are actually in the back end. So everything's kind of interrelated here. So we can see it gives us a definition of what an OncoKB is, which is a precision ontology knowledge base that contains information about the treatments and implications of this cancer. So here we see that these top 5 have a likely oncogenic effect or loss of function or and etc. So then this kind of gives us a legend of what these icons mean. And if we go into each of these tabs within this pop-up box, we can see further descriptions and definitions of what everything means. So for example, if we hover over this P10, specifically with protein change C124S, that these are some of the therapeutic implications and what kind of levels of evidence. If we click on this levels of evidence drop-down menu, it'll further expand and tell you what number 4 means. And if we click on this biological effect, it'll give us more information on that as well. And with this little black book, it gives us reference to these specific papers diving deeper into this type of variant. And so within this mutation section, this is not all that is available. So on the right-hand side, you'll see this columns drop-down menu. And so we can select all these different parameters that are available for us to check out. So I'm just going to select everything so you can see what is available. So now there's more icons. And so this little purple thing is for the specific variants. Again, it gives you more information. This fire sign is the recurrent hotspot. And again, it gives you links within that pop-up box. And so as you see, it's very interactive. A lot of things are happening when you hover over values. For instance, this HGVSG, there's a link. So when we click on it, it'll bring us to the genome nexus, and it gives us this nice little lollipop plot and more information on that, and gives us link outs to these certain resources. So there is a lot of information to explore here. And so every little bit that we hover over, it'll give us some stats on it, and it'll tell us ClinVar. So lots of information. And of course, we can download it. There's a little search bar that we can search for. So it'll just filter down for us. We will come back to something with the mutations table in a little bit. So let's move along. So if we scroll further down, there's the structural variant section. And again, they have this OncoKB. So now that we hover over it, that gives us different information with this structural variant data. And if we click on these books again, then it'll give us more details and information on them. And again, there's a columns drop-down menu. It can select and view more information there. Further down, we have the copy number alteration section. And again, you see this OncoKB icon. Any questions so far? Okay, so moving along, we see that there's this genomic evolution tab. So what this is that we can, when there's more than 2 samples, this tab appears. And so we can see the evolution change of these samples, but I don't think this is a good one. Seems like sample 1 has everything, sample 2. Sorry about that, but we can overlay the timeline, and we can see the change during that time. And there's a little tab called the heat map. So we can also see more information there. And if we hover over it, it'll give us the VAF reads for each of these specific genes. Then we have a pathways tab. And again, it's just a lot of exploration that we can click around and see. And all these visualizations we can download, and this little chart here with this cloud button with the down arrow. And here we can also download these different formats. So let's go back to the main summary view. Hold on, sorry. Let me just show you guys through a different project. So let's go here. Sorry, let me just show you something neat. So we were just exploring this tumors with TRK fusions, and I can search another project, and I'll select that by checking off this checkbox. So now you see at the top, it says 2 studies selected. So now we can explore more than 1 study at a time, a combination of how many studies you want. It does, and so we can click this button, explore selected studies. So earlier, that tumors TRK study had a total number of 76 patients. Now it is a combination of both these studies. So we can click this plus sign, and it'll tell us now it is a combined study of which of these studies that we combined. So this is what we call a virtual cohort, a virtual study. And we can share this with our colleagues if we wish. So we can click on this little bookmark icon next to the person and click share. It'll generate a link for us. And now we can share that with our colleagues or coworkers, collaborators. But say you're like, oh, I want to save this for next time though, and I don't have time right now, I want to come back. So before we never logged in, and we are able to do everything that we're up to do now. So we can log in, and we'll just sign in with Google. Oops, sorry. Oh my God. Sorry, guys. Technical difficulties. Okay. Oh my God. Yeah. Sorry, guys. This is embarrassing. What is Okay, let's just not go with this. But so what I wanted to show you was that had that worked, we would go back to our original landing page, and at the top here, instead of pan cancer, this will be further down, and the very top we will see my virtual studies, and then you would be able to see your saved study. And by clicking on that little bookmark in this field, this first text box field, we can save it whatever name. So we could have called it Combined Study MSK and the other study, and then we can save it, and then we can share it and be able to go back to it. Maybe I'll come back to this a later time and do that. Sorry, guys. So let's continue looking at this one study. So this is the project summary page. And so you can see that there are a lot of different tables and pie charts, and there's a lot of things to look at. So say you're one of those people that you're like, I don't like pie graphs. I rather look at a table of the data. So we can do that. Just go over to the graphic, and we can see that there are now 3 icons at the top right-hand corner. One of the icons is the 3 dashed lines, and we click on the first option, which says show table. And so that will now turn the pie graph into a little table for us, and it'll give us some stats. And so if we click on one of them, we can see that the study has automatically filtered down into only those with a histological grade 3. And we can further filter down the study with just those with invasive breast carcinoma. And so now we are looking at the project with just those filtered parameters, and it is shown at the top what we had done. And we can also, of course, now save this and share this with our colleagues if we wish to do so. So let's clear all that. And of course, if you would like, you can also save these and download this, although this is not a cool image to save. So maybe you want to save this Kepler-Meyer plots. And so we can just hover over those 3 dashed lines and download the data or download the graphic itself. Another interesting thing that we can do is, sorry, where was that one cohort? Sorry, let's turn this back. So we saw that we can change it from a pie chart to a table, and we can also download it. Now let's see what this compare groups button does. So it brings us to a new page. And so there's a variety of tabs that we can explore. So under survival, now we can see that there's a survival curve for those groups of histological grade. And when we hover over it, we can see for which patient ID it is, and for the percentage and some breakdown. And then we could see for each of these groups, the total number of cases, events, and the median of survival. And on the left-hand side, you can see that there's a table. We can select to show all the different parameters. And of course, you can download this. And so in the clinical tab, same idea, we can just play around and click and see the different clinical attributes. We can hover over these plots and points and see more information on which case it is, and it'll give us some more details on that. And what's more interesting is, let's go to this mRNA tab. Sorry. So here we have a table of the mRNA expression data remember, for this specific group. So if we hover over each of these items, it gives this purple highlighted thing. So we can click on it. And once we click on it, it'll generate this graphic for us, which on the y-axis, it's specifically for this gene A2ML1 of the mRNA expression. On On the x-axis is the groups again, and we can hover over it and it'll tell us the mRNA expression for each specific patient. So then in the DNA methylation, it is a similar, well, it will give us a similar thing where we have a table at the bottom and we can sort these values by clicking on the headers. And we can click on one of these entries and it will auto-populate this graphic for us. And now it is the promoter methylation for this specific gene on the y-axis and the group on the x-axis. And again, we can hover over them and see the specific values of each of these points and for what patient it is for. And anytime that you see that it is a blue, it will bring you to that patient's page so that we can dive deeper into the specific case. So that is what I want to show you here. Let's go back to this project summary page. So I also want to show you that within the mutated genes table here, we can click on each of these genes and that will auto-populate this text field at the top automatically for us, and then we can hit query. Or if you don't want to go through the table, then you can also just type it out yourself. And you can see that when I have an incorrect gene symbol, it'll tell me that it is an invalid gene symbol because cBioportal is expecting it to be an official Hugo symbol. So say I don't know what it is and I only know the HER2 protein. So if I type that in, it'll tell me it's invalid, but cBioportal is smart enough to think what you, has a suggestion of what it thinks you want. So it'll give us a question mark and we can just click on it and it'll change it for us. And now we can query it. So what happens when we hit this query button? So it'll bring us to this OncoPrint view. So this is one way to get to this. There is also another way to get to this. And so let's go back to the original landing page when we log into cBioportal. And let's find our project that we're interested. We check off the checkbox on the left-hand side of the project. And there's this other button called Query by Gene. So we can select that. And now bring us to this query page. And you can see that in this project, there are several genomic profiles. We can select all of these options and let's do it for all the samples in this project. And I have a predefined list and we can just paste it in here. And so everything is green, valid, and we can submit our query. So now this is the other way to get to this OncoPrint view. And so here there are lots of things that we can customize how we want to view things. So just by at the top here, it is profiled in mutations. So then I don't want to look at this. So then we can also change how we view things and sort how we want. So we can drag and drop these parameters. And these three little dots, if we click on them, we can also move it up and down, or we can remove the track itself. So we can just remove these. And then we can also add tracks. So there's this button called tracks. If we click on it, the first tab is clinical. So we can select what we are interested in. So let me just select a few parameters that might be interesting to us. And as you see, as we are clicking, it automatically adds it for us. So now these are the parameters that I had just selected, and we can drag and drop and move these things around. And if we scroll to the very bottom, it'll tell us what these colors mean. So it's a big legend. And so this is a little bit confusing to look at, right? So we can click on that three little dots and we can sort it. So now we can see it in a more clear way, and we hover over it, it'll tell us that this is a basal and purple is HER2. And also at the bottom, for reference, it'll tell us what these colors mean. And so within tracks, there was also another tab called heat map. And so we can add these genes to a heat map. So now that we scroll down, now it shows an mRNA expression of these scores for these specific genes. And there are lots to explore here. So every little part that you move your cursor, you can see the average mRNA score values and for which of these patients. So there's a blue link, so you can click it and it'll bring you to just those 2 patients and their profile. Okay, this one's not cool, but you get the idea, where you can hover over each of these tracks and it'll tell you which of these patients have a PR status negative or PR status positive. And we can also sort this out and view it that way. It'll tell us a breakdown of which of these samples have how much of this mutation. So it looks like ERBB2 has 17%. So we can hover over every aspect of it and we can see lots of things. So now there's this, you can see there's this field called gene panels. So if we click on it, oh shoot, what did I have? Pop-up. Sorry, there was supposed to be a pop-up. Oh, sorry, here. Yeah. So this was the pop-up. So this will tell us these are the genes in this gene panel. So that is what that means. So there's a lot of exploration to be done here. It's very interactive in this tab. And let's explore the other tabs. So you can see here, you can configure into different alteration frequencies and view the mutation data and for which cancer type categories. And this is for all the query genes. We can focus in on only certain genes if we wish. And we can play around the parameters here. This one doesn't have anything. So under plots, same idea. So at the very top, it gives us examples of what kind of plots are possible. So on the left-hand side, you can see that you can play around and choose what you want for your x-axis and your vertical axis, your y-axis, or you can swap it and it'll automatically swap it for you. And so at the very bottom, you can see that there is a legend of what these colors mean. And so we can hover over each of these points and it'll give us information and which patient it is. And we can choose another gene and it will readily generate this plot for us. Next is the mutations. Let's select this ERBB2. So now you see this lollipop plot and the mutations table at the bottom. So earlier we saw the mutation section, but that was for one specific patient and all the mutations that that patient had. Now it is another point of view where it is looking at specifically at the ERBB2 gene and all the samples that have this mutation. So all of these sample IDs are listed now on this left-hand side. And so it is in blue, so it will link us out. And if we click to it, it will be that patient's page. Oops. So it's a different point of view of looking at the mutations. And again, it is the same thing what we saw earlier. We have these OncoKB annotations, and we have these reoccurrent hotspots. And of course, if we want to see all the other parameters that's available, we can select for it as well. So at the very top right-hand corner, there is a legend to tell us what these colors mean. So if we can dive deeper. And so when we cover over each of these lollipop plots, it will highlight which of these at the bottom mutation section for us. And then at the top left-hand corner, we can add additional tracks. So let's just add all of them. And so we can see that there are these points at specific spots. And when we hover, again, it will show us these pop-up box with more information and details. For the PDB chains, if you hover over it, it'll generate a further section for us, and we can click on each one of these. And as you see, it will now create this 3D protein structure on the right-hand side. And we can change the parameters to how we would like to see this protein, and we can zoom it in and just rotate it and view it and zoom into specific areas if we wish. And of course, this can be downloaded. All the figures can be downloaded throughout cBioportal. And again, it is a research tool, so an exploratory research tool. So it's good to verify and validate on your own. So let's move on to the co-expression tab. Sorry. This is a lot of samples. So here we have the different correlated genes on the left-hand side, and we can select for each gene that we are interested, and it will automatically generate this plot for us. We can configure how we want, I guess not here, if it's positively correlated or negatively correlated, and for each specific gene to look at the mRNA expression. What's more interesting is in this comparison survival tab. Within this tab, there are mini tabs, as you see below here. If we click on survival, it is something what I showed you earlier. Under mRNA though, there is a volcano graph now, and if we select like we did earlier, it will also generate another plot, but we can see it side by side now. This volcano plot is a log 10 p-value against the log 2 ratio versus what we had previously. It was for a specific group of cases with just the histological grade, whereas now it is grouped by altered and unaltered. So when we hover over that, we can see that altered means samples with at least one alteration in our queried genes. And so we had queried earlier these 21 genes. So those are listed at the top as well for you. And so this is also another way to look at the mRNA expression. And so at the bottom as well, you can select however you want, and it will automatically populate a different graph for you. And again, we can hover over each of these plots to see which patient it is and what is the mRNA expression value. And same idea for the DNA methylation. So we can click on specific gene and it will populate that. And now it is for the promoter methylation, whereas previously in this mRNA tab is for the mRNA expression. And we can select for these different groups if we wish. So of course, again, we can select to view different information, but I guess this is all selected for us. And within the pathways, kind of like how it was in the patient page pathways, but this is pertaining to all the samples and specific genes that we had queried earlier, and we can click around and we can see the different pathways and values when we click onto these things. And under the download tab, we can download the information in specific ways that they offer and explore that way. So this is pretty much all I have today. Are there any questions? Okay.
Anyone asking?
Nope.
Okay,
Okay, hold on.
oh, there is, maybe that's me. Oh, that was
Oh.
super helpful. Okay, so you got good feedback. So before I end, and you can please ask any questions in case something last minute pops up.
Oh, I forgot. So at the very top panel, if there was something that I missed or that you want to go back, cBioportal offers lots of tutorial slides and webinar recordings that they have done, how-to videos. They have great documentation. There's also a GitHub if you want to explore that. And there is this Google group that you can ask questions or see some of the questions that other people have asked that you may want to ask as well. So they have a great resource here.
There's a question.
Since no one has a question.
Go ahead.
Unless someone else has one.
Yeah, there is one question, but it's okay, go ahead first.
I was just wondering what you kind of envision, clinicians who don't have a lot of research background using this for, like if they have a patient that they have sequence information for. And kind of what this kind of commitment is. Would they be able to do that
So there is a work in progress in trying to have clinical trials matched and to be displayed on cBioportal. That is something that's a new feature that's coming, that's being worked on. So in the future, clinicians can be able to go into their study and then their specific cases and see some of the matched ongoing clinical trials and see which patients or studies map to those. Oh, who do we contact? Oh, cBioportal. Oh, that would be me. But first you would have to contact the PI to see if you can get permission to access their data set. And then they give you the okay, then I can give you access.
Is there a way they can maybe email, or you can just Slack us? And Marianne will be happy to answer. So she's also in the panoramic Slack
Yep,
group.
I'm also in
Yeah.
the panoramic Slack group.
So that's one of the. Yeah, so that's one of the reasons why we have that platform, and is really to ask these questions, which are super quick. So if anyone wants to join and who hasn't or who isn't part of the Slack community, you can just email me and I'll add you to it. And then, yeah, so this is the end of chapter 1 of our tutorial series, which is the biologist toolkit for computational analysis. So one of the reasons why we actually did this, and I explained this before, I realized that I probably didn't highlight this too well, is really to give the tools necessary for people who do lots of wet lab and who are bench biologists. And you don't have to go through that rigor of or feel that you need to go through a computational pipeline, because there is a lot of, there's a need to bridge between what is being the output from computational analysis. I think I think it's not just me, I think a lot of people will agree that there's a need to bridge back again to wet lab biology. Without understanding our data sets, there's no way we can improve. So this whole tutorial series is hopefully to lower the threshold so that UMAP and TSNE doesn't look so scary. Yeah, they are not that scary actually, but it can be intimidating. And just to let you guys know, it's also the same for the computational biologists or the scientists. They have also reached out saying that they need that bridge back to wet lab biologists because they don't know what to do once they have a data set, how to move on. So that's where, these bridging tutorials, yeah, I should have called them bridging tutorials. Yeah, so that's the whole effort behind it. So we are going to learn from this experience. We'll reach out to you guys. We will have videos. We don't have the video for last week's Kobe session, but Paul and Emily, they're going to make an online video. We'll send it out to you guys. It will be great if you guys can also give us feedback, because feedback is where they need as well. They put in a lot of effort to build this, but without feedback, they won't know how to improve also. So it's a two-way process. Yeah, so we will repeat these tutorials in a couple of months, perhaps. But definitely this won't be the last of it. So with that, I really wanted to thank everyone for coming, and especially our tutors who took the time to come today. Yeah, I know this place was hard to find for the in-person attendees. Yeah. So with that, I will just leave it open. If anyone has any questions, you can ask. If not, have a good afternoon. And we have our grant writing workshop tutorial this Thursday. So feel free to sign up for it. Thank you. And there is food and drinks for those who attended in person. Anyone? No one. Can I stop sharing? All right. Anyways.
There's a lot, how we manage to do